आप सभी को मेरा सादर प्रणाम, नमस्कार व मंगलमय सुप्रभात। Today I am going to present the series of this biochemistry lecture. Now definition of biochemistry already over. So today I am going to present the history of biochemistry. In this series, we are starting the whole biochemistry history as milestone work of the biochemistry. Here, first, Carl Neuberg in 1903 first used the term biochemistry. Who used the term biochemistry? So first Carl Neuberg used the term biochemistry. Carl Alexander Neuberg was an early pioneer in biochemistry, often referred to as the father of biochemistry. He is also known as the father of the biochemistry. Who? Carl Neuberg. Carl Neuberg is known as the father of biochemistry. He defined biochemistry as science which describes the structure and functioning of living organism in the language of chemistry. Okay. Now, Alexander Neuberg worked on the solubility and transport in the cells. He also worked on the chemistry of carbohydrate, photochemistry, as well as investigating and classifying different types of fermentation process. He was also a pioneer in the study of the chemistry of amino acids and enzymes. Okay. Now, Antony Lavoisier, he is also known as the father of modern biochemistry. Who is the father of modern biochemistry? Antony Lavoisier is also known as the father of modern biochemistry. His classical research on oxidation and the role played by the oxygen in the process led him to investigate burning in the body, and he concluded that oxygen is consumed in the reaction, the carbon dioxide is eliminated, and heat is evolved. Now we also we must know about the Bresh Chandra Guha. Professor Bresh Chandra Guha is also considered as a father of biochemistry in India. He is a distinguished fellow of the National Institute of Sciences of India. Okay. Dr. Guha was honored for his contribution to science and for his services to the country. He had achieved eminence by his extensive work on the biochemistry of vitamin C, vitamin B complexes, and in food and nutrition. Guha joined the Bengal chemical and pharmaceutical works and initiated there many lines of work on the preparation of vitamin concentrate and biologically active compounds from the glandular products. Okay. Now, Frederick Euler in 1828 by synthesizing urea, an organic molecule and a waste product of animal metabolism, from ammonium cyanate, an inorganic molecule obtained from the mineral sources. Okay. Many science historians, this in vitro synthesis of urea by Euler, known as the recognized as the starting point of biochemistry. Okay. Now, in 1878, German physiologist William Cohen coined the term enzyme, which comes from Greek word enleven to describe this process. The word enzyme was used later to refer to non-living substances such as pepsin, and the word ferment used to refer to chemical activity produced by living organisms. In 1897, Edward Buchner began to study the ability of yeast extract to ferment sugar despite the absence of living yeast cell. In a series of experiments at the University of Berlin, he found that the sugar was fermented even when there were no living yeast cell in the mixture. He named the enzyme that brought about the fermentation of sucrose, gymase. In 1907, he received the Nobel Prize in Chemistry for this for this biochemical based research work and his discovery of cell free fermentation process. Okay. Now, in 1926, James B. Sumner showed that the enzyme urease was a pure protein and crystallized it. Sumner did likewise for the enzyme catalase in 1937. The conclusion that pure protein can be enzyme was definitely proved by Northrop and Stanley, who worked on the digestive enzyme pepsin, trypsin, and chymotrypsin. These three scientists were awarded the 1946 Nobel Prize in Chemistry. The term metabolism is derived from the Greek word metabolisms, for change or overthrow. The history of the scientific study of metabolism is from 400 years. The first controlled experiments in human metabolism were published by Santerio in 1614 in his book Ars de Statica Medicina. This book described how he weighed himself before and after eating, sleeping, working, sex, fasting, drinking, and excreting. He found that most of the food he took he was lost through what he called insensible perspiration. Now, in 1665, Robert Hooke had successfully invented the microscope. Because of this discovery, Robert Hooke was the first to examine what a cell appears to be close to. His description of the cells was published in Micrographica. However, the cell walls observed by Hooke did not indicate the nucleus and other organelles found in most living cells. Antony Philip van Leeuwenhoek was a Dutch microbiologist and microscopist in the golden age of Dutch science and technology. A largely self-taught man in science, he is commonly known as the father of microbiology and one of the first microscopists and microbiologists. Van Leeuwenhoek is best known for his pioneering work in microscopy and for his contribution towards the establishment of microbiology as a scientific discipline. The German biologist Theodor Schwann is considered a founder of this cell theory. He also discovered pepsin, the first digestive enzyme prepared from the animal tissue, and experimented to disprove spontaneous generation. In 1838, Schwann became familiar with Mathias Cylinder microscopic research on plants. Schleiden described plant cells and proposed a cell theory in which he considered to the key to the plant anatomy and growth. Schwann not only verified the existence of cells, but he traced the development of many adult tissues from early embryo stage. Louis Pasteur was a French chemist, pharmacist, and microbiologist renowned for his discovery of the principles of vaccination, microbial fermentation, and pasteurization. His research in chemistry led to remarkable breakthroughs in the understanding of the causes and prevention of the disease. He is regarded as one of the founder of modern bacteriology and has been honored as the father of bacteriology and the father of microbiology. Okay, here why are we talking about the microbiology, bacteriology, and others cell theory? Because these are the parts of the biochemistry. Because if we are not going to talk about this, then our history of biochemistry is incomplete. So this is necessary to talk about all these things. Jonathan Frederick Messner was a Swiss physician and biologist. He was the first scientist to isolate nucleic acid in 1869. Messner also identified protamine. Messner had isolated various phosphate-rich chemicals which he called nuclein. Now this is known as nucleic acid from the nuclei of the white blood cells. In 1999, in 1919, Foves Livians, a Russian physician and chemist, first discovered the order of the three major components of a single nucleotide: phosphate, pentose sugar, and nitrogenous base. He was also the first to discover the carbohydrate component of RNA, ribose, and the carbohydrate component of DNA, deoxyribose. In 1937, Hans Krebs discovered the process of the citric acid cycle, also known as Krebs cycle, in honor to him, which is a series of chemical reactions that occur during cellular respiration. Glucose and oxygen convert to water, carbon dioxide, and energy. This is the whole Krebs cycle. The aim of this Krebs cycle to generate the energy. In 1953, he received the Nobel Prize in Physiology or Medicine for his discovery of the citric acid cycle. In 1944, while working on bacterial sample, Oswald Avery first suggested in 1944 that the genetic material of the cell was possibly the deoxyribonucleic acid. Oswald Theodore Avery was one of the first molecular biologists and a pioneer in immunochemistry, but he is best known for the experiment that isolated DNA as the material of which genes and chromosomes are made. In 1950, a scientist named Irving Chargaff began concluded that almost all DNA, regardless of organisms or tissue type, still maintains certain properties even as its composition varies. He postulated the Chargaff rule, which say that the amount of cytosine is equal to the amount of guanine and the amount of thymine is equal to the amount of adenine. The discovery in 1953 of the double helix, the twisted lattice structure of DNA by James Watson and Francis Crick, marked a milestone in the history of science and gave rise to modern molecular biology, which is largely concerned with the understanding how gene controls the chemical processes within cells. Their discovery yielded ground-breaking insight into the genetic code and protein synthesis. This is the whole of the my history of the biochemistry. Here we cover major major steps, major milestone discovery of the in the field of the biochemistry. So